Good morning, guys. If you haven't done so already, please let me know in the comments that you are here for the day. Looks like most people have done that already. Heads up, you should have received back all of your assignments at this point with marks and comments. So if you look them up on the assignments tab, you should be able to see what your mark is on all your assignments. If you owe me something, that is, there is an incomplete assignment, or you haven't written one of the quizzes. Remember, there are two. Some people were surprised when I communicated with them, but yes, there have been two quizzes. Please make sure that you're completing those as soon as possible. They basically have to be done today, if there's any chance of me updating things before I send out your reports on tomorrow. So unfortunately, if I don't have everything from you, I'm not really able to give you a mark tomorrow, which doesn't really look very good. So let's see if we can get that figured out. Anyway, not many people in that boat, but there are a few. So if you want to chat with me about that, feel free to send me an email. But some people are not in a great place regarding that, which is unfortunate, but it does happen. You're probably wondering, what is my overall mark in this course? Great question. I'm going to post those today. Again, keep in mind that if I don't have everything from you, you won't have a mark posted there. It'll just say I or insufficient data. I can't remember if I was going to say something else, but I lost my train of thought. Before you hand in any of your assignments for this unit, which we're going to, well, I guess potentially start doing tomorrow, because tomorrow is the last day of the unit. Please make sure that you have a look over the comments, especially from your vocabulary lists. Some of you guys are doing a great job. You got detailed descriptions that include an explanation as to why the particular term is important in that unit. You provide examples when it makes sense to provide examples. A lot of you are doing that already, which is great. But if not, I've probably left a comment with regards to that on your vocabulary list. So take a look at that. From this point forward, I'm not going to say that I mark harshly, but certainly for the first two, you don't really get much feedback before you get you don't get any before you hand in the first two. All you have is the instructions. And so I'm pretty chill about, being missing components, like, oh yeah, sure, you didn't connect it to the themes. I mean, it's not a huge deal. I remove maybe a mark or something for that. I mean, depending on how egregious it is. But kind of from this point forward, everybody knows what the expectations are. Like, those are the things you need to include, and if you don't include them, then your mark is commensurate to not including the things that you need to include in your vocabulary list. So consider that. The other thing that I noticed with regards to if you're going to be discussing, for example, why a term is important in the particular unit, think about what you're actually saying in that sentence where you're justifying its importance. Are you saying, basically, if you were just to simplify what you said, are you saying it's important because you need to know it to understand it? That's not really explaining why it's important. That's just saying it is important twice. So think about that. When you're giving your justification for why a particular term is important, think about why we learned it. Think about it. This is the whole idea behind this, is, again, to help you synthesize the actual knowledge. You have to think about why it's important to know that. Why is it important to know about phenotypes? Okay, well, that unit is all about gene expression. It's about how genes, or the molecular component of information, is converted into physical components in a organism. I'm just speaking off the top of my head here, which is basically what you're going to be doing in your definitions as well, with some editing, obviously. But so we need to understand the concept of a phenotype, because this unit is all about how particular molecular components of your genome are presented physically in the organism, which is a phenotype. You know, like you think about like the actual use of the term and like why it's important to know it within the context of the unit. So just keep in mind that you're not. This isn't a lot of people, but certainly some people have been. Their justification for each term has been just like, you need to know this because like it doesn't make sense unless you know it. I was like, well, that's true. I mean, you could say that for every single term, but why is it important in this unit to know about that? And honestly, if you can't think of any reason why it's important to know a particular term, it's probably not core terminology and doesn't belong in your list of 30 terms. Like the idea is that you're picking 30 that are core, that are important to know to understand the stuff from a particular unit. And I should say that it should be fairly easy to find 30. Most of these units have a ton. This unit included. There's probably a lot of words that are new to you, I would assume. So, and then that are also important to know in the context of the unit. So I don't see any questions from the content from Friday. Let me pull up the content page here. We are on day 13. On day 12, we did a little bit about the anatomy of the respiratory system, and we looked at the mechanics of breathing and some illnesses of the respiratory system. So what can happen when the respiratory system kind of fails in various ways. What you guys are going to be working on for the first little bit, it's in the same notes package. Scroll down here. So after the illnesses portion here, there is a little section on lung capacity. So there are various ways to measure the capacities of the lungs. There are various aspects to your lung capacity that can be measured, and they mean different things. And so, for example, certain parts of your lung capacity, certain components of your lung capacity increase when you become more physically fit. Certain components of your lung capacity are always the same, no matter what. Certain parts of your lung capacity will change depending on if you have an illness. Okay, so knowing a little bit about these is very useful. I'm going to get you guys to read the lung capacity section in your notes. It's on page 443, and it's going to go through these terms right here. They're also sort of summarized. This is the exact same figure from the textbook, and it sort of helps as a visualization of what you're actually measuring when you measure these different components of your lung capacity. So I put that there on the bottom as for reference for you as well. It's helpful for understanding exactly what you're talking about, and you guys are going to define these terms. Okay, I mean, ideally, whenever you define something, you're using your own words, but I mean, in this case, it's going to be a hybrid of what you're reading in the textbook. So you're going to start there. Okay, so we're going to be that's sort of like the very end of the respiratory system unit. We're just going to talk about these various capacities of the lungs. All right, so that's the beginning. There are also some questions, which are right. Oh, there's a short reading in the textbook. It's section 10.3. Section 10.3, that's slipping my mind. I don't remember what that is. From a textbook. Yes. So section 10.3 talks a little bit about the idea of partial pressures. So this is sort of important in understanding what is pushing oxygen across the membrane. We talked about diffusion, and we talked about some things that affect diffusion. You have to have a thin membrane to allow gas to diffuse across it. We talked about if you increase the concentration on one side of the barrier, it's going to increase the rate of diffusion through the barrier. That's basically if you keep ventilating, you're going to keep the concentration of oxygen high in your lungs, which means that there's going to be more transport into your blood. So this chapter is going to talk a little bit about the idea of partial pressures, which is the concept that the different parts of air, the different partials of air, are going to be contributing different amounts of pressure, of the total pressure. And so, for example, if something is 20% of the air, like oxygen, 20%, then whatever pressure there is outside, like let's say that the pressure is, what do you guys measure pressure in chemistry? Kilopascals? Or do you use bar? I can't remember. Anybody taking chemistry? Kilopascals is what you use, right? Or do you use millimeters of mercury? There's so many different pressure measurements in science. Bar is the imperial system, so it's not used a ton in metric countries. But I don't remember what you guys use in science. Kilopascals? Anyone taking chemistry? When you learn STP in chemistry, standard temperature and pressure, what's the pressure that you learn for STP? 101.3 kilopascals? I haven't taught chemistry in like 10 years, so I don't remember this. Just pascals. Okay. All right. So at sea level, 101.3 is considered to be okay. All right, cool. Yeah. So 101.3 kilopascals is considered to be like the base temperature at sea level. And so the idea of partial pressure is that if you want to figure out the pressure of any particular gas, and you guys may have talked about this in chemistry, so there's some crossover here. Where's my pencil? If you know that the total pressure is 101.3 kilopascals, if you want to know, and that you know that it's 20% oxygen, what that means, it's going to be slightly more than 20. I should be able to probably do that math in my head, but I'm not gonna. Then the actual pressure that oxygen is exerting in that situation is 20.26 kilopascals. And that's important because the actual total amount, this number, is what is going to be determining how quickly oxygen diffuses into your lungs. So when you go up in altitude, let's say you're going to go and climb Everest, okay? This doesn't change really. The amount of oxygen as part of the total, 20%, is basically the same at the top of Everest. It's not that there's actually less oxygen total in the air. The oxygen amount is still 20%. So the concentration is pretty much the same for all of this level of the atmosphere, and it goes up quite high. But the other number is going to change. So that's this number. So when you get to the top of Everest, that kilopascals number, the total pressure, I think I have it written here somewhere. It's somewhere around 30, I think. I bet you it's in this chapter in the textbook. I can't see it, but it's somewhere around 30, okay? Which obviously is much lower. You still have 20%. Ooh, still have 20% of it in the air. And so now the total pressure that's pushing oxygen into your blood is something around 6 kilopascals. That is an extremely low pressure, considering that when the blood actually returns from your body, once you've used up most of the oxygen in your blood, there's still something like 5 kilopascals of pressure left over in your blood. Okay? So remember, it's the difference, it's the gradient between the two sides that actually forces oxygen over the barrier. So if you've got your blood returning from the body. Okay, so you've got blood here coming back in. I'm going to use blue to signal deoxygenated blood, although it's not actually blue. So this is blood passing through in the capillary, and then on the other side we've got our alveoli over here. If you've got 20 kilopascals on this side, whoa, what just happened? Sorry. If you've got 20 kilopascals over here, which is standard at sea level, and you've got roughly 5 on this side, because it's not going to be none. Not all of the oxygen is removed from your blood by your cells. There's still some left over. This is a big difference between the two of them, and so you're going to get a lot of diffusion of O2 going across here. But if you've only got 6 kilopascals over here, let's say that you've gone up in a really high altitude, like to the top of Everest, and you've got 5 kilopascals returning, this is only going to be a teeny, teeny, tiny little bit of oxygen that is going to go across here. And in fact, the amount of diffusion that you get across your lung membranes at the top of Everest is so small that if you were to spend any significant length of time there, more than a day, you're going to end up with brain damage. So it's not, you can't live at the top of Everest. It's not a livable environment. If you want to take a couple steps at the top of Everest, just take a couple regular steps. You have to basically take like 20 or 30 breaths per footstep that you take at the top of Everest, which is ridiculous. It's extremely difficult to breathe at the top of Everest because of this very, very small difference in total pressure of oxygen across the membrane. And that's why it's called there's like a name for that zone. If you go like above 7,000 meters, it's called like the death zone. Or no, 8,000 meters it would be. Above 8,000 meters, it's probably in the chapter. It's in the chapter somewhere in the textbook. It's like the death zone or the dead zone, because at that point you cannot get enough oxygen in. So what do people do? Well, you can't change this number at the top of Everest. You can't change that. It's going to be low pressure, unless you're climbing Everest in a diving suit that's under pressure, that's not a thing. You can't do that. So what you can do is you can change the other number. You can change this number by actually bringing supplemental oxygen with you. So what people do is this. It's still 30 kilopascals. Okay, that total pressure, you can't change that. That's the ambient external pressure at the top of Everest. But the second number here, you basically just turn into, I don't know, something like 90%. It's 100% oxygen that you're breathing, but it's not a perfectly sealed system and stuff like that. So you're not actually getting 100% pure oxygen when you're climbing Everest, but it's a much higher number. So when you do this, even though this number is lower, times 0.9. Now here you're going to get maybe 27 kilopascals. So there's a way to maintain the partial pressure of oxygen by breathing supplemental oxygen as you go up. And it's not a perfect system. You got to carry the cylinders up, which they're heavy, although you can pay a bunch of Sherpas to carry them for you. But anyway, pretty much everybody does that. There are very, very few actual recorded, what is it called when you get to the top? Summiting. There's very few summiting events of unassisted people that don't use supplemental oxygen. I don't know how many there are in recorded history. Not many. And basically no one does it because it's extremely, extremely difficult. So you pretty much got to use supplemental oxygen because doing it like this is very problematic. Now, I should mention that when you're up there, when you're sleeping and stuff like that, you can't be like breathing 100% oxygen all the time when you go up, because it's just impractical to always be on 100% supplemental oxygen. You just can't carry that much of it up there with you. So you don't end up being on supplemental oxygen the entire time, which means that yes, the potential for brain damage still exists up there. So anyway, that's the thing. Anyway, so that's what this chapter is all about. I just wanted to give you a quick summary because people struggle a little bit with that idea of partial pressures, especially if you're not taking chemistry. Those of you that have already taken chemistry, like the idea of partial pressures, you probably already covered in class. And so you have maybe some background in that already, but I just wanted to mention it. And then there are some questions that go along with that reading, chapter 10.3. Anyway, it's a fascinating chapter. It talks about a bunch of stuff, mostly about altitude. And then the last component of it, it talks a little bit about the neurological control of breathing, which is also a fascinating topic. So that's the end of chapter 10.3. So the questions that go along with this are page 451, numbers 1 to 6, and there's a little EXA quiz that goes along with it as well. That's for practice. Then, once you're finished that, and that probably won't take you too long, we are going to start our last system of the unit, which is the circulatory system. To give you a little bit of an introduction, we're going to start with a really cool documentary. I watched this documentary even when we're in class, mostly because a lot of the visuals that are in this documentary are amazing. There are things that I cannot show you. I don't have any method to show you some of the stuff that's filmed here. For example, they have a number of shots where they have sagittally sliced some blood vessels, or well, I guess this tissue with blood vessels, and they found some samples where you can still see the entirety of the blood vessel, like from end to end, and they pump blood through it. So it allows you to actually see red blood cells going through capillaries. I mean, that's really fascinating to actually be able to see that happening under the microscope. I can't offer you that in class. So there are a number of demonstrations like that in this documentary. It's not super long. It's 25 minutes long. There are some questions that go along with it. Again, these are just to make sure that you're kind of following along and that you're picking up the things that you should be picking up while watching the documentary. But so that is the first part of the new notes package. So the document is called The Human Pump. So there's that. And then in the afternoon, we are going to go through the first part of this note together. We're going to talk about the components of the circulatory system, including blood and what's in blood, a little bit about the immune system and white blood cells, and then we are going to get into the parts of the heart. Normally at this point in the course, we would do a dissection of a heart, but it's not in the cards this year, unfortunately. So that one's not going to happen, but I will we'll discuss the parts. I will say at the end of this unit, I'm not able to get a fetal pig or go to a fetal pig at the moment. So I do have footage of my fetal pig dissection from last quad, which I've edited down to a much more reasonable size, and I have that video. I'll give you the option to watch it if you're interested. So we will do that, but it's not quite as interactive as I'm hoping, but well, we'll make the best of it. And then the last thing that I'm going to get to today, that dissection thing is tomorrow. The fetal pig dissection is tomorrow. But the last thing I'm going to get to today is I'm going to talk about the 3 types of blood vessels, how they're different from one another. Those are arteries, capillaries, and veins. And then tomorrow, we're going to talk about the control of your heart, so how you actually keep your heart beating, and how you're able to speed up and slow down the rate of your heart. We're going to talk about the electrical signals that happen inside of your heart. You've probably seen an EKG read before, at least in a movie or something like that, the thing in the background that goes boop, boop, boop. So we're gonna talk about that, what exactly you're looking at when you look at an ECG or an EKG. And then we're just going to talk a little bit about blood pressure and why blood pressure measurements are important. The last component tomorrow of this unit that we're going to discuss are illnesses of the circulatory system. Just like we did with the respiratory system, we're going to talk about some important illnesses of the circulatory system. And then that is all. You have the option to watch the fetal pig dissection if you like. It's kind of cool. It's fairly easy to see what's going on in there. My camera is actually not bad at showing fairly good detail inside the fetal pig, so you're welcome to watch that. It's a pretty good review of the systems, which is why we do it. And then that's it. Then we're going to work on the project, the medical technology assignment, and we'll be done with this unit. Hooray. Any questions about what you're working on now? So just to very quickly recap here, you're going to read that lung capacity component from the book, or from the notes, and then from the text. It's only one page. It's page it's set on the sheet, I think. Nope, not this sheet. Anyway, it says on the lung capacity sheet, which is page 6 of the respiratory system notes, it says which page it is in the textbook. I don't remember. There's a short reading, chapter 10.3 in the text. You can add any new terminology that you find to your vocab list, and then there are some questions to go along with it and an exit quiz. When you're done that, there is a little documentary on the circulatory system. It's an introduction to the circulatory system, and then I will see you guys at 11:05. I gotta post your marks now, by the way, too. See you in a bit. Hey guys, if you haven't done so already, please let me know in the comments that you were here for the second half. Okay, so first of all, question 3 on page 451. I'm gonna go through that together, and then I gotta dig up the answers to the worksheet. I do have it, and so I'll post it on Brightspace, probably in the same spot on the video. I thought it was up there already, but maybe I forgot to include it. Let's see. Worksheet. I did forget to include it. I will add that in, because I do have that. I wonder if I have it in my notes, completed notes package. It might be in that. Circulatory system. No. System part 1. Huh. Well, I have it. Anyway, I'll find it. I'll add that to it. It'll take me a minute. I've got it in my USB key, I'm pretty sure. All right. So what question for question 3 on page 451 is asking? It says gas exchange occurs in 2 locations, the alveoli in the lungs and at the body cells. Using the concept of partial pressure and pressure gradients, explain the gas exchange. So the idea of the pressure gradient is what we've already been talking about. That's just diffusion. The idea that things always move from a high concentration to a low concentration. That's partial pressure. Partial pressure is a concentration measurement. So basically it's saying that, I mean, you could pick any gas you wanted, but if you have a membrane that's permeable, so it has holes in it that allow things to go from one side of the membrane to the other, and then you have a high concentration over here, and those square brackets just mean high concentration, or they just mean, sorry, square brackets just mean concentration, and you have a low concentration over here, you're going to get movement going from high concentration to low concentration, just naturally through diffusion. That's what we were talking about before the break when we looked at that idea of like the little balls that were bouncing around, right? If you got lots over here and few over here, they're going to equalize across the barrier. They go from high to low concentration. So all this is saying here is describe the two circumstances that exist in the lung and at the cell for why you have movement across the membrane. So we can just focus on oxygen. So I'm going to visually describe this as I'm talking about it here. I'm going to draw it as I'm talking about it, but you could just write this out verbally as well. You wouldn't necessarily have to draw anything. But okay, if you've got an alveoli, you've got a certain concentration of oxygen inside the alveoli, and you've got your blood vessel, which is running next door to it. Okay, so if you want to know the partial pressure of oxygen, which is the concentration, it's just a measurement of this. Okay, you take partial pressure is equal to the total pressure times the percent makeup of that particular gas. Okay, so if the total pressure, let's say that this is at sea level. I mean, we're not at sea level, we're a little bit above sea level. I could get out a barometer and measure the pressure here. I don't know what that pressure measurement is, but it would be slightly lower than what it is at sea level. But let's assume STP, okay? So 101.3 kilopascals. This is what you use in chemistry all the time. I realize, again, we're not at sea level, but we're moderately close to sea level. This isn't really like a mountainous area here, so this is going to be fairly close. And then times the percent of the specific gas, and it's about 20%. And then we just did that calculation, but of course I forgot what the number was. 20 or something or other. $20.26. Okay. That is the kilopascals. That is the partial pressure of oxygen in the atmosphere. Okay, or you could think of it as the concentration of oxygen, the pressure of oxygen, how much oxygen is pushing on everything around it at the surface. Okay, so this is going to be roughly, this is going to be roughly the partial pressure of oxygen in the alveoli. It's not quite that high, because remember that you're always removing oxygen from the alveoli into the blood. It's diffusing across the membrane. And so, and you got to ventilate to continuously replenish it. So it's going to be slightly less than that number. Okay, so maybe it's 19 or 18 kilopascals. It's less than what it is atmospherically because you're removing some all the time. Okay, but sure, it's approximately atmospheric, especially if you're breathing regularly. It's going to be roughly close to atmospheric pressure, okay? And you've got now blood that's coming through here from the body, and it's already dropped off a lot of the oxygen that's contained with it. And you know what? Actually, I can look this number up. I can look up the partial pressure of oxygen in venous blood. Like, give me 2 seconds. I'm going to actually just Google that. That's pretty easy to find. It says between 30 and 40 millimeters of mercury. Let's say 35. I don't know what that is in kilopascals, but I'm going to find out. So 35 millimeters of mercury in kilopascals. 4.6. There you go. That's an average. It's not going to be exactly that, but a little high, a little low. Not really particularly important for our purposes, but what was the number that I just said? 5.6? 4.6. So that's pretty close to what I had written there before. I think I said 5. But sure, let's say that it's 4.6 kilopascals. So if you have a differential in pressure between the two of these, you're going to get an equalization across the membrane, right? That's diffusion. So because there's this difference, oxygen is always going to be moving in this direction. And the greater the difference between the two of them, the more is going to diffuse across the membrane. That's the gradient. So the gradient is basically just a difference between two locations. I think it talks about gradient, right? It says use the concept of pressure gradients. So a gradient just means that there's a difference between two, and then the greater the difference, the higher the gradient, the more movement you're going to get across the membrane. So this is a fairly big difference between the two of them. It's about 15 kilopascals. And so you're going to get a lot of movement. Okay, and that's the reason why you have to keep breathing, because if you stop breathing, this partial pressure, this quantity of oxygen, is going to slowly start decreasing. 20, 19, 18, 17, 16, and the longer you hold your breath, the less diffusion you're going to get across the membrane, because this number is going to go down and down and down, which is why you have to keep breathing, because you want to maintain this as close to the atmospheric partial pressure as possible. Got to keep breathing. Okay, so that's what's happening at the alveoli and why oxygen is going into the blood. Okay, so then as this diffuses across, it's going to increase the partial pressure in the blood. Now, it doesn't increase it all the way up to 20 kilopascals. It gets somewhere in like the 18-ish range. Okay, so it's not gonna get all the way up to atmospheric partial pressure, which just makes sense. It's not going to fully equalize as it's passing by, but it does increase quite a bit. Okay, and so then this is basically going to become your arterial blood then. So let's say now you're sending that oxygen. There's 18 kilopascals of pressure. Now I should mention I'm using kilopascals here because that's probably what you used in chemistry for measuring pressure, and you can absolutely use that as a measurement tool. That's fine. Now, but in biology, we typically use millimeters of mercury for measuring pressure, and millimeters of mercury is just a different pressure measurement. And I could look up the conversion real quick. I don't know what 1 millimeter of mercury is, but I will look it up. So 1 kilopascal is 7.5 millimeters of mercury. Okay, that's the conversion factor between the two of them. And so you could just interchange them. In your textbook, I think that you're typically going to see millimeters of mercury used as a pressure measurement, not kilopascals. In biology, we tend to use millimeters of mercury. But then again, when you're taking atmospheric measurements, like you're just out there with a barometer taking atmospheric measurements of pressure, typically you measure in kilopascals, because that's sort of like a chemistry measurement. And so there's obviously an interaction between chemistry and biology happening here. So I'm not really sure which one is correct to use here. In my physiology courses, all throughout my entire education, we always use millimeters of mercury in any biological context. So I should probably use that here, since almost everything is going to be written in that. But anyway, it doesn't matter. It's just a different unit for the same thing for a pressure measurement. All right, so now let's imagine that this blood is going to circulate, and it's going to make it to a cell. Okay, so we've got our arterial blood coming by here with 18 kilopascals of pressure. I'm having such difficulty writing kilopascals. And then we've also got a cell. Okay, so the cell is just where we're dropping off, and it's got a little nucleus over here, and we're going to drop off some oxygen to the cell. Now the cell's not necessarily touching the capillary. There's also going to be cells that are like over here, that are maybe like a little bit away from the capillary, and in that case the oxygen is actually going to diffuse into this space in the middle, which is the intracellular space. So there's actually some space in between the cells, and there's a fluid in there, intracellular fluid, which is more or less continuous with the blood. So that liquid goes into the blood, and there's liquid coming out of the blood going into the intracellular spaces. And so the oxygen, to get to a more distant cell, is actually going to be just diffusing its way across like this. Obviously, it has less access to oxygen as the cells that are directly adjacent to capillaries. But that's good enough for most cells. You're going to be, as a cell, you're going to be within 3 cells of a capillary, pretty much no matter where you are. And depending on the type of tissue, you may be closer. So in muscle tissue, which is extremely oxygen dependent, there's actually more capillaries in the tissue, and so fewer cells are further away. But in things like your skin and stuff like this, there's usually a little bit more distance in between because they don't have quite as high of an oxygen requirement. So anyway, if you need to do an exchange here, you've got 18 kilopascals of pressure of oxygen in the artery. And then inside the cell, it's using up oxygen, right, to do all of its, because its mitochondria are taking it in and producing ATP. And so you're going to have a lower concentration in here. You might have like 3 kilopascals of O2 pressure in here. And so obviously if there's going to be that significant a difference between the two of them, then you're going to get diffusion of oxygen going from the artery into the cell. Okay, or from the capillary, I should say, into the cell. And then after this point, the blood's going to keep flowing, but it's going to have less. So maybe it's going to have, well, what did we say when it returns? 4.6 kilopascals. Okay, so it's going to give it up most of its partial pressure, and now it's got a lot less oxygen in the artery, a lot less partial pressure of oxygen, and it's going to circulate around through the veins, through the venous system, and make it back to the lungs again. Hopefully that cleared that up. For the quiz, it says complete it before Monday. Oh, did I write that? I think that might be an error. Sorry, give me 1 second. I might have forgot to update that. Does it say that on the content tab, Kim? Oh yeah, it does. That should say complete the quiz before, for you guys, it would be Thursday. Midnight on Thursday. Let me update that. Because that would be the day after the final day of the unit. Thursday, May the, and I'll just include the actual date and that makes it easier, the 27th. Thanks for pointing that out. Fixed. Okay, so hopefully that answered, I don't know who asked that question. Michaela's question? Michaela, did I do okay there? Did that make sense? Hopefully. All right, let's get to what we're going to discuss this afternoon. So anyway, I will post this. Give me a minute. I'll do it after after we've gone through the note together, because I've got to have a look through my USB key. It might take a few minutes, but I will post it. So we're going to proceed through this information here for the circulatory system. It's actually in a presentation, which I have posted as well. So if you want to look at the presentation yourself, there it is. It says I'll be using a slideshow for the note. I've linked a copy of it here. So you guys are welcome to look at this yourselves as well. It's accessible to you. Okay, so a circulatory system. If you had an opportunity to watch the documentary, it does a great introduction. If not a big deal. You can watch it during Learning Block 3. But there are some visualizations in that documentary that are really cool, especially watching the red blood cells go single file through the capillary. I think that's awesome. So anyway, there's some cool stuff in there. I highly recommend it. There are 3 components to a animalia circulatory system, and most animals have a circulatory system of some kind. Sea sponges excluded, obviously, and actually peripera, which are things like what are those things called? Jellyfish and medusas and those types of animals don't really have a traditional circulatory system. They operate basically just by diffusion. So their tissues are in water, and the water has oxygen, and the oxygen just diffuses into their tissues. It's a little bit more complicated in sea sponges, but it's essentially the same thing. Most more complex animals have something akin to these three things in them. If you really want to nail it down, you can say that this applies to all chordates, but it does apply to most other groups of animals, not just chordates. So what are the three things you need for a true circulatory system? You need a fluid that transports stuff around. In mammals and in chordates, we call that fluid blood. In insects, for example, in arthropods in general, they have something similar to blood called pseudolymph, and it also transports gases around, but it's not exactly the same as blood, and it doesn't usually have hemoglobin, although there's something similar to hemoglobin in some arthropods. Anyway, we don't need to get. Let's focus on chordates. Okay, so chordates have blood. They have blood vessels, which are tubes for moving the blood about inside of the body. And again, other groupings don't necessarily have tubes. Arthropods have a cavity that they just push blood around in. They just kind of fan it around inside of a cavity, so it's not really a blood vessel, but chordates have blood vessels. And a heart, which is a pump, something to move the stuff around, the liquid around. Again, other organisms like arthropods and annelids, which are roundworms, they have something like a heart. It's usually called a heart, but instead of it being a closed pump like our heart is, their heart is more like a fan that just kind of waves the blood around in an area. It doesn't really pump it per se, it just kind of fans it around. So, which I guess is sort of a pump, but anyway, we're going to be mostly focusing here on chordates. And you know what, more specifically, when we talk about the anatomy of the specific components, this mostly applies to mammals. So why don't we just, in order to make this a little simpler here, I'm going to say that this is almost exclusive to mammals rather, okay? Which you are a mammal. There are variations of these things in the other chordates, but they are variations. They look slightly different than what we're going to discuss here, but the basics are more or less the same. Okay, so but this stuff specifically applies to all mammals. So why do you need a circulatory system? Well, you got to move food around. We've already talked about the idea of metabolism. Well, in order to do metabolism, you have to get the stuff to metabolize into your cells. So you need to get sugar. We also talked about the idea of protein synthesis and genetics. Well, how do you get all the stuff for that in there? Blood. So blood is to move the amino acids around as well. Fats, which we also use as fuel. So that stuff is being transported in the blood to your cells. You got to get the other stuff necessary for metabolism, which is O2, to your cells, which we were just talking about with the respiratory system. That's how you get it in the system. And then how you get it to your target is through the circulatory system. And you got to remove the waste products from metabolism, which is CO2. You got to remove waste, so all of the breakdown products when you're cycling through protein, like ammonia, uric acid, urea, those need to be removed, and removed from your body as well, which is done in the kidneys. The circulatory system is used to maintain your temperature, and we talked a little bit about pH and respiratory acidosis and alkalosis, but it's used to maintain your body temperature and the pH. The body temperature thing is kind of interesting because you might have talked about this in grade 10, but when you sweat, what you're basically doing is you're taking the liquid part of your blood, the water part, and you're just pushing it out of your body. Because where did that water come from in your sweat? It was your blood. So that's kind of weird. You don't really think about that. It's obviously not red because the red blood cells are not not being pumped out. It's only the water component, but your blood is coming out. Kind of. Parts of your blood are coming out when you're sweating. It's used to fix leaks. So that would be to clot wounds. We're going to talk about that process in more detail. And you have white blood cells in there to destroy invaders. There are also the other chemicals that you use to communicate between your cells, and those are being transported around. We talked about the idea of hormones and what they do. They're types of proteins used for communicating between cells. They are transported in the blood. And antibodies, that's sort of part of the immune function. Okay, so that's a lot. The circulatory system is pretty important. In a mammalian circulatory system, there are 2 circuits that the blood goes on. So they're separate from each other. The blood goes on one circuit, and then it goes on the other circuit, and then it goes back to the other one, and then the other one, and it keeps switching between these 2 circuits. And the intent of that is that one of the circuits is for getting oxygen and dropping off CO2, and the other circuit is for giving away oxygen and picking up CO2. So they are completely separate. It's why your heart has 4 chambers. So the 2 sides of the heart are basically independent from each other, although they're electrically connected, but they are, in terms of where they're pumping, are separated. So you've got your pulmonary circuit. Pulmonary means of the lung. So you're going to the blood is going to take this pulmonary route and get its O2. Then it's going to return back to the heart again on the left side, and it's going to join the systemic circuit. So then it's going to leave the heart and go around through the systemic circuit to all the different tissues of the body, drop off its oxygen, pick up the CO2, come back to the heart on the right side, and then it's going to go through again and go through the pulmonary circuit. Okay, so 2 separate circuits in action here, a pulmonary circuit and a systemic circuit. The right side of the system deals with deoxygenated blood. That's blood coming back from your body. And the left side of the system only deals with oxygenated blood, and that is the blood that is coming back from the lungs, that is full of oxygen. If I'm going too fast at all during any parts of this, please let me know, and I will slow right down. Okay, let's talk about what's in the blood first. But before we get into blood vessels, and we are going to go into blood vessels, let's talk about the little components here that you find in your blood. So your blood is made up of, oops, drop my pencil. Your blood is made up of plasma, white blood cells, platelets, and red blood cells. Let's talk about the percentages that you find of each of these. So if you were to take a sample of blood, we used to do this lab in grade 11. We're not allowed to use human fluids anymore. Things have changed a lot since I started teaching. But we used to take a little blood sample from you, not with a syringe. You only need a single drop of blood to do this, and some capillary tubing. So it would just be like a finger prick. And we had these little devices that were single use that just prick the tip of your finger, and then you toss it out, and then you put a capillary tube on top. And it sucks. You might do this lab in university. A lot of university labs still do this. The capillary tube, using capillary action, which we're going to talk about in the plants unit, is going to pull the blood up into the glass tube, and it's a teeny tiny little glass tube, so it doesn't take much volume to fill it up. Then you put it in a centrifuge and you spin it around for a minute. Doesn't take very long. And what the centrifuge does is it takes all the heaviest components and it throws them to the bottom, and the lightest components float up to the top. Okay, so basically separates things by density. So the most dense part is going to be the erythrocyte component. And that usually makes up about 44% of your blood. Erythrocytes, by the way, are red blood cells. So a cyte just means like a cell, okay, or a type of cell. And erythro is the scientific name for red blood cell, erythrocyte. If you are an elite athlete, you probably have a higher percentage of your blood as red blood cells. Oh, I shouldn't say elite athlete. Even if you're in good shape, cardiovascularly, you probably have more than 44% of your blood. If you were an elite athlete, you might have something like 55 or 60% of your blood is red blood cells. because that's one of the things that gets enhanced when you train. But you, even if you're moderately active, you probably have more than 44%. Now, that being said, if you're extremely inactive, it might be the opposite. So you might have a very low percentage of red blood cells, maybe 40% or even in the 30s. If it gets too low, obviously, it becomes problematic for you to stay alive. So as long as you're relatively healthy, it doesn't get too low, but it does fluctuate a little bit. So there's a range. The reason is that red blood cells are responsible for transporting oxygen, which is what your muscles need to function well. So, that's the bottom component there. The buffy coat is the part in the middle. It looks like just like a little tiny white line. It's less than 1% of your blood volume, and that is where you find your leukocytes. It's mostly leukocytes are white blood cells, which have the immune function, and we'll talk about a little bit more in a second. And platelets, which are used to seal any leaks. The platelets make up a very, very small percent, even of the buffy coat, and so you can't really see them. They're in there, but it's mostly white blood cells that you're seeing. And the last component is basically water. Plasma is mostly water, but this is what you would sweat out when you sweat. So looking at these things in a little bit more detail, if you look at the plasma component, which is on top, it's mostly water. There's a little bit of protein in there, albumin, globulins, fibrinogen, regulatory proteins. Those are all different types of proteins that are cell signaling and immune. They have an immune function, and you could take a whole course in this, so I'm not going to get into too much detail in terms of what they do, but there are a number of proteins in there. And then there are some other things that are dissolved in there. Electrolytes, which would be salts, sodium, and potassium. Other nutrients like dissolved sugar, respiratory gases that are dissolved like carbon dioxide and oxygen. Some of that is just dissolved in the water component. And waste products that'd be like urea, uric acid, stuff that your cells are trying to get rid of, ammonia. Little teeny tiny amounts of that in your blood as well. But you can see the plasma is mostly water. When you are in the hospital and you're having your blood volume replaced, let's say you've done a lot of bleeding, if you're simply dehydrated, they're mostly going to give you plasma in the hospital. Now, it might be plasma from a person, or it could be artificial plasma. Artificial plasma is basically salt water. I mean, it's saline solution, really. Although you can get versions of artificial plasma that have some of these components in them that are synthetic versions of that. And then that would be if you were replacing a lot of volume. Usually you can just get away with using saline, which is basically salt water, if you're not having to add tremendous amounts of volume. If you've bled a lot and you don't only need to replace the liquid, but you actually don't have enough oxygen transporting capacity in your blood, then you'll need to get a full transfusion of blood, not just of plasma, because what you need is this part right here. You need the erythrocyte component. Those are the red blood cells, and they are used for transporting oxygen. So then you would require a full transfusion. Now, if you know you're going to need a lot of blood in advance, the best blood to give somebody is your own blood. So for a lot of surgeries that are likely going to require a fair amount of transfusion afterwards, you usually go in a month prior or a couple months prior and give your own blood, which they will store for you. And then your body will make more red blood cells. And then after the surgery, they can give you back your own blood. That's the safest way to do it, because obviously your own blood is not going to be incompatible with your body. In general, and this is going back to the blood type stuff from the previous unit, if you guys remember blood typing from genetics, as long as you get the right type of blood, you usually don't get too many complications from getting a blood transfusion. That's the main thing, that the blood typing is the main thing that causes complications. However, it is possible to have an immune reaction to other things in somebody else's blood, the albumins, the globulins, other things that might be slight variations that are not recognized by your immune system. So it's not a completely risk-free procedure, unless you're getting your own blood transfused back, in which case it's relatively risk-free because it's your blood, so your body's cool with that. Like I said, there's a teeny tiny amount here that's platelets. We're going to talk about their function in a second. They are used for sealing leaks. And then you've got these big ol' cells that are used for immune function. That's going back, way back when we talked about how vaccines work. We mentioned that there are cells that eat bad guys, invaders. Those would be these guys in the middle, the monocytes. There are also cells that produce antibodies and that are used for recognizing invaders. Those are the things that are going to interact with vaccines. Those are lymphocytes and neutrophils. I'm not going to get into the details of what all these different ones do because, again, you could take a whole immunology course on this in university, and I'm certainly not going to cover it in any requisite amount of detail necessary there. But anyway, they have various immune functions. They're used for identifying bad guys and building your defenses, essentially, to those bad guys. That's really all you need to know for this course. I don't think I realized that I went through these one at a time. Did you guys already get this information from the previous slide? Because I'll jump through this quickly if you did. This information is on the previous slide, I believe. No comment. Okay, I'm going to assume you did. Oh, it's just a little small. Okay, cool. Then I'll hover here for a second so you can fill this information in. But we already discussed the plasma, so. I mentioned that red blood cells are called erythrocytes. You make them in your bone marrow. They are one of the few cells you have in your body that don't have a nucleus, and so they have a fairly limited lifespan, at which point they are absorbed by your spleen, and those materials are redistributed. But you do cycle through them fairly quickly. They have an interesting shape. It looks kind of like an inner tube, although there's no hole in the middle. That type of shape is called a biconcave disc. And they are obviously for transporting oxygen and, to a lesser extent, carbon dioxide. A lot of the carbon dioxide gets dissolved in your intracellular fluid in the plasma component. But they also transport CO2. If you've ever heard of people blood doping in sports, for example, and this is maybe a bit of an archaic reference now, but Lance Armstrong, is that still a name that is like a known name? Or did that happen too long ago now that no one has any idea who Lance Armstrong is anymore? 7 or 8-time Tour de France winner. After a long investigation, finally admitted that he was blood doping. So the way that Lance Armstrong was blood doping was that he was taking a hormone called erythropoietin. And erythropoietin is the hormone that your body produces that tells you to make more red blood cells. It's like a switch that says, make more red blood cells. So that's how Lance Armstrong was blood doping. He was taking erythropoietin. And you can test for erythropoietin, for exogenous erythropoietin. Exogenous just means from outside of your body. It's slightly different chemically than endogenous erythropoietin. Endogenous means from inside of your body. So you can test for it, but he had a very elaborate strategy for avoiding testing after he had taken erythropoietin. And you need to catch it pretty quickly after it's taken. Otherwise it has a fairly short half-life, so it doesn't exist in your blood for very long, and it's not really detectable after a fairly short amount of time. So he managed to get away with it for 7 or 8 seasons. Now, according to him, and I fully believe him in this, elite cycling is absolutely full of cheaters. I'm sure it still is, which is kind of sad. I think it's harder to cheat now than it was 5 or 6 years ago, but it's still full of cheaters. There's such a strong pressure to cheat in cycling because it's at such an elite level. You really have to have every part of your physiology maxed out, and even in order to even have a chance at competing, which is really hard to do. I mean, you could be training 7 hours a day every day, and still not be elite enough to make it. Even after training for years and years and years, you could have the best trainers, and you just still can't break that like peak in order to get to the elite level. And of course that's a huge force for people to cheat, which is basically just like, well, if I can't do it and I'm putting in everything I got, there's only one way to go. So anyway, I shouldn't say that everybody's cheating at the elite level, but I have a feeling it's still fairly common. There are lots of other ways to cheat other than taking EPO, so we'll save that conversation for a different time. Not every sport is like that, but I have a feeling it is in cycling. Anyway, hemoglobin, that is the cool protein that you have at the center of each red blood cell. It is made of 2 heme groups. Each hemoglobin can carry 4 oxygens, one at the center of each heme group. Sorry, there's 4 heme groups. Each one can carry 1 oxygen. So you've got 1 oxygen in each heme group, and there's 4 heme groups. So that's 4 oxygens per hemoglobin. And when you bind hemoglobin to an oxygen, it changes the way it reflects light slightly, which makes it bright red. And this leads to the most often misunderstood thing about blood, which is that oxygenated blood that is bound to oxygen, is red, and deoxygenated blood that is no longer bound to significant oxygen, that would be what you have in your veins, is blue. People say that all the time. People think that, but it is most certainly not blue in the veins. So I just want to point this out because this is one of those things that is like a general misunderstanding that exists. If you just ask somebody about this on the street, I would say 70% of people you ask will get this incorrect. So I just want to show you the difference here between oxygenated and deoxygenated blood, so you can see the difference in color. I'm sure I could find a picture of these. Yeah, there we go. Perfect example. So you'll notice from looking at these, there's oxygenated on the left, which is a bright red color, and then deoxygenated blood is on the right-hand side here. I guess you could call it purplish, although it's really just darker red. Certainly you would not call that color blue. No one would call that blue. So when you look at your veins, which are running fairly close to the surface, and we'll talk about why that is later, but your veins run fairly close to the surface. So when you're looking at your arm, all the blood vessels you can see are veins. They look blue. And the reason they look blue is more about the layers of tissue that are sitting on top of the vein and which colors of light those layers of tissue are absorbing, and the colors of light that the blood inside is absorbing. And when you combine all of those together, you get blue light being reflected. But it's not really blue. So if you were to actually look at the blood that's traversing through your veins right now, you would say it's basically just dark red. So anyway, let's get that confusion taken care of. It also doesn't help that anytime you look at a circulatory system, like look at this stuff, and anytime you look at a circulatory system diagram, they always use red to indicate oxygenated and blue to indicate deoxygenated. It's in basically every diagram of the circulatory system, which is very confusing. I think to the average person, when you look at these, you're like, well, yeah, it turns blue. Like, it's in every single... Look, you can't even find a diagram where they don't do that. It's basically in every circulatory system diagram. And I think that has just become like a convention that you just draw deoxygenated blood as blue, even though it's not blue. It's never blue. So I just want to point that out because that is confusing, that it's always drawn as blue. But in real life, it is not blue and never is blue. Let's talk about white blood cells. I mentioned this already, this idea that they are patrolling the inside of your body. They're not only in your circulatory system, so they also travel through your lymphatic system. And they also travel through the spaces in between your cells, the intercellular spaces. There are white blood cells that just move about in there. Now, it's difficult for them to get around in between your cells, and that's why if you need them to be there, like you have an infection or an injury, and you need white blood cells to be there, you get inflammation. Inflammation is swelling in a tissue, and what that is basically pumping the area in between your cells full of fluid. And the reason that you pump the fluid in there is so that the white blood cells can get to the site, wherever it happens to be, the injury or the infection. So that's what the purpose of the inflammation is. But inflammation has its own hazards. So they're moving around in there looking for bad guy flags, as we talked about previously. And if they find them, they mark them, usually for a different type of white blood cell to eat. And you can, that's a really cool picture at the bottom there, which is the different types of white blood cells under the microscope. That's a pretty good optical microscope right there that took those pictures. I've certainly never used an optical microscope that had that kind of resolving capacity, so cool. But yeah, they can move around in between your cells, and they can travel in and out of the circulatory system in order to get the bad guys, whatever they happen to be. They also produce a bunch of enzymes that can potentially destroy invaders. They could be taken inside of the white blood cell, and then it makes a bunch of enzymes that break them down. And they make antibodies, as we previously mentioned, to fight off infection. We talked about how that works with regards to vaccines. Platelets are basically cell fragments. I'm not sure you'd call them cells. They don't have all the components of regular cells. And they're used for blood clotting. They don't have a nucleus, so they're similar to red blood cells in that regard. I guess they are cells. They don't have a lot of the components of a regular cell, including they have no nucleus. They don't live very long either. They have an even shorter lifespan than red blood cells. So let's talk about how the clotting mechanism actually works. So when there is a leak, okay, in your circulatory system, and notice there is a leak down there due to a push pin, I don't know, whatever causes a leak. And this could actually just be from knocking one of your limbs against something hard. It doesn't even have to be very hard. You're going to get tiny leaks popping into your capillaries and stuff like that, even just from minor bumps and stuff like that. Usually you repair them extremely quickly, but you get them all the time, just from little bumps. What happens is you're going to rupture some cells when you do that. And your platelets can read, when you break a cell open, basically its contents just get shunted into the tissue around. And those chemicals are typically not in that liquid, right? That's just, it's basically just like a cell's exploded and all of its parts are just floating around. Those proteins and shapes are not usually there. And the platelets can detect that. They're kind of patrolling around in your blood, and when they detect those chemicals that shouldn't be there, that are only there when cells break, they activate this process. So the platelets will begin to convert a hormone, or they begin to produce a hormone called thrombin. Okay, so your platelets are producing thrombin at the site. They aggregate at the site. So they're able to navigate and stick to the site of injury. And what thrombin does, it's another hormone. There's often cascades of hormones that are involved here. Thrombin will take a protein, which is just floating in your blood, which is called fibrinogen. Fibrinogen is just floating around in your blood. And you can think of fibrinogen as kind of like a piece of a spider web. That's really what it is. It's like these little tubes that are similar to just these like wispy pieces of spider web. And normally they are not sticky. So fibrinogen is not a sticky protein. It's a piece of spider web that is not sticky. But when it comes into contact with thrombin, the thrombin kind of changes its shape just a little bit and makes it sticky. And once it changes shape, it's called fibrin. Okay, so fibrinogen is converted into fibrin, and fibrin will begin to stick to the platelets that have already aggregated at the site. So the platelets basically lay down the foundation. They go to the site of injury and stick, but they don't have the ability to block the gap on their own. They release thrombin, and the thrombin will convert that fibrinogen to fibrin, which is basically, again, it's like a sticky piece of spider web. And then it's going to form a webbing, the fibrin, over the hole. It's going to build a patch over your hole. And as long as the hole is fairly small, there's a picture of it right there under an electron microscope. So we've got the really dense areas here are actually platelet cells. So we've got a platelet cell there, there's a platelet cell there, there's a platelet cell right there. And sticking to it are these wispy fibers, which are fibrin. It was fibrinogen, but it was activated by the platelet to make it sticky and turn it into fibrin. And so the fibrin is now sticking and forming this web, which is, as you can see in the diagram right there, actively blocking the movement of red blood cells. It's creating a net that is preventing the leak. As long as the leak is relatively small, this process works fairly well. So if it's a small leak in a couple capillaries, or I don't know, a lot of capillaries, but it's relatively small, you can be fairly confident that your body is just going to block that off with this process. Now, if you've got a big bleed, or it's under a lot of pressure, because maybe it's arterial or something like that, going to be a little bit more of a problem. So what you need to do in that circumstance is you usually need to apply pressure, and the pressure prevents a rapid loss of blood through that wound. And then what you're doing is you're giving time for your body to start building nets over the larger holes that have been punctured into your circulatory system. So that's why you're applying direct pressure to a wound, to a bleeding wound, to provide an opportunity for this to occur. Now, if the wound is in an artery, it's under extreme pressure, and we'll talk a little bit later about how much pressure is actually in the artery. It's a lot. You may require a little bit more than just applying pressure to it. So that you may require some type of surgical repair if it's an artery that you've damaged. It typically isn't, though, and that's because your arteries are usually very deep in your tissue. So this is a really cool example of all of the small blood vessels that are found in the face. Your face is highly vascularized. It's one of the most vascularized areas of your body. If you've ever blushed, then you've experienced a rapid increase in the circulation to these blood vessels. Why is the face so highly vascularized? Good question. There are a couple of theories. They probably have something to do with the fact that blushing it used to be an important communication mechanism between humans. And it is in other primates. Although we don't really use blushing for important communication any longer, and so it's probably not particularly important for us now. However, from an evolutionary perspective, it used to be important. Therefore, we evolved to have this unique high vascularization of the face. Anyway, that's just as a little bit of an aside, but this is an actual person. You may be wondering, what the heck? How do you get something like this? Well, it's from an exhibit called Body Worlds. If you've ever gone to see Body Worlds before, it is I don't know what to call it, art exhibit, but it's made with real bodies. They're mostly donated, I think. And what they do is they plasticize. They use this special chemical to turn specific tissues into plastic. And then they use acid to eat away the other tissues, or they're surgically removed. I don't know if you call them surgically anymore. They are removed, debrided, using tools. And what's left over are these people with certain parts of their physiology or anatomy preserved. It's all kinds of weird stuff. So the people are put into like positions like they're playing tennis or whatever, and it's usually people that are like they've removed the skin or they've made particular organs visible, or sounds a little bit macabre. It is a little bit macabre. There used to be an exhibition of this at the Science Center when I was in Teachers College. That's a fair amount of time ago, but it tours around. I wouldn't be surprised if it came to town again. Anyway, this is the thing. If you're ever interested to see what is out there in these Body Worlds exhibits, you can Google it. I think I just spelled Body Worlds. But if you look at a Google image search of these are all different parts of the Body Worlds exhibit that travels around. It's pretty crazy. If you're interested in anatomy, it's fascinating. It's not only humans either. There's a whole bunch of different animals that are on display as well. Got a bull. There's a person riding a horse, and all of the things that you're seeing are real. That's an actual horse, and that's an actual human. Fascinating. There was some controversy when this exhibit was touring that some of the bodies that were used were actually Chinese prisoners that were executed. However, my understanding is that is not true of the exhibits that tour in North America. So, not necessarily applicable to these. I obviously think that is not cool. It's actually terrible. But I don't think that any of the exhibits... these are mostly donated. So you can donate your body to Body Worlds, and they'll plasticize it and turn it into a work of art. If you're interested, I don't think a lot of people do that, but as far as I'm aware, all the ones that toured in North America are donated. These are donated bodies. So, oh, really? Sorry, crashed. Oh, that was the last page anyway. So that brings us to this. Cool. Brings us to the heart. Any questions about anything we've covered so far in terms of the structure, the anatomy, the physiology of the respiratory system, circulatory system rather? We're going to talk about the anatomy of the heart now. And this is where we're going to finish off. The next part here is all about and blood vessels. And tomorrow we're going to get into the electrical activity of the heart. A lot of the heart stuff that we're going to talk about is actually review from grade 10, or it should be review from grade 10. I covered this in my grade 10 class. Not every grade 10 teacher covers exactly the same anatomy. The curriculum book is a little bit vague on what exactly you're supposed to cover, so kind of pick and choose. Okay, let's talk about the heart. I think you probably already know that it's a pump. That's what it does, pumps blood around. And it's attached to a complex network of vessels, which we're going to talk about in a little bit more detail shortly. It pumps about 70 beats per minute at rest. If you are highly fit, it probably pumps significantly less than that. Could be 60 or 50 beats per minute, or even lower if you're an elite athlete. And that's because when your heart is very fit, it actually pumps more blood per beat. So it doesn't have to pump as many times per minute in order to properly perfuse your body with blood. But my resting heart rate is 70 beats per minute, almost exactly. I looked it up on my watch a minute ago here, which is about 100,000 beats a day, and that's normal. So it's pretty important that it keeps doing that. To keep you alive, you got to keep transporting that oxygen. With every beat that it pumps, it pushes fluid through about 100,000 kilometers of vessels. And keep in mind that we're talking about vessels that are only one cell thick, your capillaries. And what you just saw, what just the capillaries of the face look like. And so you can imagine how, if you were actually to lay them end to end, that's a pretty long tube of blood vessels. 100,000 kilometers is nothing small. So there are a lot of vessels running through your body. Remember that cells are no more than 3 cells away from a capillary at any given time. So when you add them all up, it's a lot of length. So the heart's got to be pretty strong. You got to pump blood through a lot of tubing. It needs to be flexible in terms of how much blood it delivers, because when you exercise, and we'll talk about how you control your blood, your heart rate tomorrow, but when you need more blood, you got to be able to turn the thing up to maybe 220, 260 beats per minute. There's a range of maximums. The maximum, the average maximum, is somewhere around 220 beats minus your age. It goes down slowly as you get older. So for me, my max heart rate is probably somewhere around 190, 195. I think that is actually true. When I'm exercising at maximum capacity, it's probably somewhere around 195 beats per minute. It can be a little higher or a little lower depending on the person. Yours is probably somewhere around 210 or something like that. Again, average. That would be its maximum amount of beats per minute. There are 2 circuits that you pump to. One is to the lungs, and remember that's where you're collecting O2 and dropping off CO2. And then it goes through to the body cells, where it's going to distribute those things, distribute O2 and pick up CO2. So I'm gonna proceed through the heart here. I'm going to start on the right-hand side, and I'll go through these one at a time. So if you're desperately trying to scribble these down, I'm going to go through them one at a time here so that you can write them down. So on the right-hand side of the heart here, you have blood arriving from the body. So this is blood that is deoxygenated. It's had its oxygen used up. There is a big old vessel at the top that collects the blood from the top half of your body. And that vein is called the superior vena cava. That, along with the bottom version of this, are the 2 largest veins in your body. So the superior vena cava, superior just means above or from above. And obviously it's above, going above your heart. So that's why it's the superior vena cava. Vena just means vein, and cava just means cavernous. Cavernous just means it's big. So it's the superior big vein, okay, or the upper big vein, okay, if you want to translate that into modern English from Latin. And the bottom one here is the inferior vena cava, coming from the bottom half of your body. Inferior means lower, vena means vein, and cava means cavernous or large. So it's the lower big vein, just in Latin, inferior vena cava, or cava. Okay, so you've got blood coming from the lower half of the body in the inferior vena cava and from the upper half of the body in the superior vena cava, and they are going to both come together in this smallish chamber on the top right-hand side of the heart. Keep in mind when I'm saying right-hand side, I know you're looking at this diagram and being, this is the left of the diagram, right? Yes, it is. But imagine that we are looking at a person. Okay, so the person is facing you as you look at them, and this would be the right-hand side of the person who you are facing, okay, which is why it's called the right side of the heart. It's on their right-hand side. It's on your left, but their right, okay? That's why this is called the right-hand side of the heart, even though, obviously, as we view it from the outside, it's on our left, but it is on the person's right-hand side, This seems to be a big confusion among students for some reason. So that's why the right hand stuff is on the left side of the diagram and the left side stuff is on the right side of the diagram, because the person is facing you. Okay, so all the blood collects in the right atrium. It's not very strong and it's very thin, because the only job that the right atrium has is to pass blood down through this big old valve and into the right ventricle. So the right ventricle is going to do the majority of the pumping on the right hand side. The right atrium doesn't really do that much. It just helps fill the ventricle up to the top. And the valve in between those two things, the big valve, I have it highlighted in pink here, that valve is called the atrioventricular valve, and because it's the one on the right hand side of your heart, it's called the right atrioventricular valve, or AV valve. The reason that you have a valve there is because when the right ventricle pumps, you don't want blood to go backwards into the veins again. Obviously, if it went backwards, that would defeat the point of the pump. So you got to keep the blood moving in one direction. So the valve only allows blood to come into the ventricle, but it doesn't allow blood to go back out of the ventricle through the same way that it came in. Okay, that's a valve. It's like a one-way door. So then the right ventricle is going to pump that deoxygenated blood. Oh, I wanted to mention, I have this highlighted on here, but you have these really cool little strings. And if you did a heart dissection in grade 10, you might have seen this. I really wanted to do one this year, or at least this quad, but it doesn't look like we're going to get back into school in time. Anyway, what I've just highlighted in yellow there, those little strings, those are your heart strings. Yes, you do have heart strings in real life. They are called chordae tendineae, and their job is to prevent this valve, this AV valve right here, this valve right here, from prolapsing, from going backwards. Essentially prevents the valve from breaking. So it's attached to all these strings that keep it from going backwards. And those are called chordae tendineae. They're extremely strong. When we did do our heart dissection, you could pull on these things almost as hard as you can. And you can't really break them. They're very, very strong. And keep in mind that they have to be like that for your entire life. They can't break, or your valve will prolapse. So they have to be pretty darn strong. There's quite a bit of pressure in there. So then the right ventricle contracts, and it pushes blood up through this little valve. And again, the valves are always to keep blood moving in one direction. That little valve right there is called the tricuspid. I have it written over here on the left-hand side. The reason it's called a tricuspid is because it has 3 flaps on it. 1, 2, 3 flaps. And those flaps are called cusps. And there's 3 of them. So it is the tricuspid. The 3-cusp valve. Tricuspid. There is a similar valve on the other side of the heart, but it only has 2 flaps, so that is called the bicuspid. Bi meaning 2. So blood goes up through the tricuspid and out through these 2 arteries. There's one artery going out in this direction, pulmonary artery, and then there's another pulmonary artery going in the other direction. It's not really, you can kind of just see it in the background. It's this one back here. And that's because the blood has to split up and go to the left lung and to the right lung. So the word pulmonary means of the lung. And so the pulmonary artery is the artery of the lung. Most of these Latin phrases that are like explaining what things are, or that are like the names of these things in anatomy, make sense. If you know what the Latin words mean, they're not like just like random words or anything. They usually indicate exactly what the thing is. Pulmonary means of the lung, and artery means really, an artery is a vessel that is traveling away from the heart. So pulmonary would be the lung version of the vessel that travels away from the heart, pulmonary artery. So the blood is going to go to the lungs, get its oxygen, drop off its carbon dioxide. That's what it does. And then it needs to come back to the other side of the heart. Now we're on the left-hand side of the heart, which is primarily for oxygenated blood. And it's returning in a vein. Veins are vessels that are coming towards the heart. So we've got a vein here from the lungs, which is pulmonary. So that's the pulmonary vein. And there's two of them. You can see the two of them there, one on the top and one on the bottom, because one is coming from the left lung and one is coming from the right lung. And they're going to enter, again, that little sac on top of the heart called the atrium. But now we're on the left-hand side of the heart, and so this is the left atrium. Again, the atriums don't do a lot. They help fill up the chamber below, which is the ventricle, but they don't pump a ton. They're pretty thin. If you look at how thin the tissue is up top, like you actually look at how thin it is, it's pretty thin. Whereas if you compare it to how thick the tissue is of the ventricle down here, it's the big honker. That's a very thick muscle because it's got to pump really hard. So the left atrium fills up the left ventricle, or it helps fill it up. It's going to go through a valve, just like it did on the other side, in between the atrium and the ventricle. That's this valve right here, which is the AV valve, the atrioventricular valve, but this one is on the other side, so it's the left one. And then the left ventricle is going to pump. If you compare the left ventricle, by the way, to the right ventricle, you'll notice that it's actually thicker. So if you look at the thickness here of this and the thickness here of this, the left ventricle has a lot more meat to it because this side of the heart has to pump to the entire body. It's not just pumping to the lungs, which are right beside the heart. The left-hand side of the heart has to pump to the top of your head, the bottoms of your feet, the tips of your fingers. It's got to pump really far away, and so it's got to put it under a lot more pressure. And if you actually measure the pressure leaving the left ventricle, it's quite a bit higher than the pressure that's going to the lungs, again, because it's got to go further. You got to push harder to get that flow. So then the blood is going to go up through this little valve. That valve is called the bicuspid. There, I got it labeled at the bottom down there. And I mentioned already that the bicuspid is called that because it has two cusps. So a cusp on each side, 2 cusps. Bi means two, bicuspid valve. And then it goes out to the strongest and thickest artery in your entire body. That's what's at the top right here, and that blood vessel is called the aorta. So you'll notice that there are 3 large branches coming off the aorta on top, one to go to your head, one to go to your left arm, and one to go to your right arm. And then it circles around. You can see that it's circling around the heart and actually comes out at the bottom behind the heart. It's not shown on here, but I kind of just drew it in there with an arrow. And that goes to the bottom half of your body. One more part that I forgot to mention, but is kind of important. There is a big barrier in between the two sides of the heart, right here in the middle, which we're going to talk about when we talk about the electrical activity of the heart. And that part is called the septum. Now, when you're writing the quiz and when you're doing stuff for this unit, you don't have to memorize these. Obviously, you can open this document at any time and look at the parts of the heart, but we are going to reference them quite a bit, and so it's a good idea for you to know what the parts of the heart are. Traditionally, in this course, you would have to memorize these. I get you to label it on a test, but I have no way to assess that any longer. So, congratulations, you don't have to memorize the parts of the heart, but they are good to know. And if you do know them, we're going to be referencing them constantly, especially in the next lesson. It will make it a lot easier to understand if you know what they are and what they do. So, just as a heads up. Those are the parts of the heart, or at least of the mammalian heart. All mammals have the same layout. Other animals, other chordates have a similar layout. For example, birds have something similar to this. It's not 4 chambers, and so there is a little bit of mixing happening between the left and the right hand side here, and it's not quite exactly the same, but it's similar. So, okay, last thing I want to get to here, and then I'm going to let you work on the questions, the last part here. I want to talk about the difference between the 3 vessels. Okay, so there are 3 major vessels. We already talked about this idea. You got your arteries, you got your capillaries, and you got your veins. Okay, so the most important thing to know about each of these is their main function. What do they do? So the artery is always going to carry blood away from the heart. So if blood is going away from the heart, it's in an artery or an arteriole. Arteriole is just a smaller version of an artery. Okay, it's like artery light. That's the arteriole. If it's in between, so it's the teeny tiny vessel running through the tissues and dropping off gases and picking up gases, basically doing all the work of the system. That's the capillary. So the capillaries are for actual exchange. Okay, if anything is being exchanged, it's going through a capillary. Blood that's traveling in arteries isn't actually exchanging gases with the surrounding tissue. It's moving too fast. It's under too high pressure. The vessel is far too thick. So it's not actually allowing stuff to go in and out of the vessel. So that all that stuff happens at the capillary, not in the artery. And then finally, once the blood has traveled through the tissues and now it's collecting on the other side and making its way back to the heart again to go on the circuit again, that's the job of the veins. So veins are always going to be carrying blood back towards the heart. The key here is towards the heart. If a vessel is carrying blood towards the heart, that's a vein. So if you look at these in detail, if you zoom in on an artery or a vein, the structure of them is a little bit different. So it's important for you guys to know what the key differences are structurally here between arteries and veins. And they're obviously associated with their function. So first of all, and I'm going to start with arteries, and to some extent arterioles. Remember, arterioles are just smaller versions of arteries. The job of the artery is to transport blood under high pressure to the various parts of the body. That's what it does. It's sending it's a high pressure hose, is what it is. So when you think of what does a high pressure hose have to be in order for it to work properly, the most important thing it has to be is strong. Because if it's not strong, it's going to spring a leak. So when you look at the actual structure of it, you will see that there is a thick layer on the outside of connective tissue. And that connective tissue, that's the, oh, sorry, it's not drawing on here. Why is it not drawing on here? This part right here, this thick layer on the outside is connective tissue. And that's just to hold everything together. It would sort of be like if you've ever used pressure hosing, like with an air compressor or something, it's got to have like a strong plastic basically on the outside or rubber to keep the pressure in. That's the connective tissue's job. So that's on the outside and it's pretty thick. Below that is a muscular layer, and it's also quite thick. And the reason that you need that muscle layer is to direct blood to different places. So you can vasodilate, which means relax the blood vessel. So if you vasodilate and make the vessel really big, more blood is going to go through the vessel. That probably just makes sense to you. If you make the hose bigger, more blood goes there. So for example, if you're running, all of the arteries that go to your leg muscles are going to vasodilate and allow more blood to be transported to the legs. Same thing is true that you want to direct blood away from certain areas while you're exercising. For example, you don't want to direct blood to your digestive system. You don't want a whole bunch of heavy blood going through your digestive system when you're trying to run. So the exact opposite happens to the blood vessels in your digestive system when you exercise, which is that they vasoconstrict. They get smaller, which restricts the amount of blood going to your digestive system. So you can redirect blood flow using this muscle in your blood vessels. And that's why it's got to be so thick, because there's a lot of pressure. So in order to change the size of that vessel as there's pressure flowing through it, they have to be pretty strong. So there's a lot of muscle and a lot of connective tissue in these. There's also a fairly thin layer on the inside, an endothelium, which is just basically a protective layer on the inside to protect the muscle. This is the layer that gets damaged if you have high cholesterol, It leads to atherosclerosis. We're going to talk about that more in the next tomorrow. But it's a protective layer on the inside. One last thing I want to mention about the arteries is where they actually go in your body. So they are found deep. Arteries, because they're under such high pressure, about 3 psi, by the way, that's the amount of pressure that's in like a properly inflated football or soccer ball. That's a lot of pressure. So if you pop that, if you rupture an artery, blood is gonna come out of it very quickly. And so in order to protect your arteries, you've evolved your arteries to sit very deep in your tissues. Virtually none of them are near the surface of your body. There's only a couple places where your arteries are close to the surface, and I'm going to show you where they are. Get up here. These are the places, by the way, where you typically take a person's pulse. Oh, am I not going to be able to find this? I looked this up last quad and it was easy to find. So, maybe I'm using the wrong search terms here. So, oops. Surface. Close to the skin, close to the surface. Here we go. Here we go. They're also known as the pulse points, because this is where you can check your pulse. You got to put your finger on it and you can actually feel blood pulsing through the artery. Other than these few points, the arteries are typically running quite deep in the tissue, basically in the center, and that's to avoid damage. So you've got your maxillary ones, which are on the outside of your jaw, your carotid ones in your neck. You've got your brachial artery, which runs in the little sort of diamond of your elbow, on the inside of your elbow. Your ulnar and radial, which are at your wrist. You got the femoral, which is on the inside of your thighs. You've got the popliteal, which is in the little diamond on the back of your knee. And you've got the dorsalis pedis. And the posterior tibial too, but that one is not quite as close to the surface. But you can use both of those to take your peripheral arteries in the leg. And so other than those sort of few small points, your arteries mostly run quite deep. I should add that diagram to my note, actually. That's a good diagram. Anyway, so they're generally found deep. On the other side of things, if we look at the vein, and the venule, by the way, is the smaller version of the vein. Venules come together to form veins. So, I mean, they're structurally, they're very similar, but a venule is just a smaller version of a vein. So if you look at their structure, first of all, there is connective tissue on the outside. However, if you compare it to the artery, you'll notice that it's a lot thinner. Why is it a lot thinner? Because it's not under high pressure anymore. Remember, this is after the blood has gone through all of the capillaries, dropped off the gases, and is just kind of loosely collecting on the venous side. So there's not a lot of pressure on this side, and so there's just not a lot of connective tissue because you don't need to hold it together quite as strongly. There is a little bit of muscle, so there is a muscular layer on this side as well, but you don't do nearly as much redirection of blood flow on the venous side, and so it's much thinner than the muscle on the arterial side, because you just don't need it to be very thick. And then you also have an epithelial layer, or an endothelial layer, which is on the inside, and it's relatively thin, and just like it is in the artery. So the function of that is fairly similar. The one major difference that you're going to see here, other than the thickness, is that there are valves on the venous side. And we already talked about the function of a valve. Valves are there to make sure that blood always flows in one direction. On the arterial side, you don't need valves. And the reason why you don't need them is because it's under really high pressure. You're never going to get blood going backwards in an artery because there's a strong pump forcing blood to always go in the same direction through an artery. It's never going to go backwards. But on the venous side, because the pressure is so low, you actually need valves built into your veins to prevent blood from backflowing, going backwards into the capillaries again. You want to keep the blood always moving in one direction. So how the blood typically flows here, it's so annoying. This thing is starting to crash on me. 1 second. So how the blood typically flows in a vein is that muscle action and movement of your body is going to push the blood up a little bit, and then it's going to kind of fall back onto a valve, and then it's going to get pushed up again and fall back onto a valve, and then it's going to get pushed and fall back onto a valve. So it's this repeated push up, fall back, push up, fall back, push up, fall back. That doesn't need to happen on the arterial side because it's under high pressure, but the veins basically keep everything flowing in one direction to ensure that the blood makes it back to your heart. So there are valves on the venous side. Okay, they're also running quite shallow. So all of the blood vessels pretty much that you can see, actually this isn't showing up very well on the camera, but I've got big veins here in my forearm. All the blood vessels that you're able to see from the surface are pretty much all veins because they run fairly close to the surface. They're under low pressure, and so if you do happen to rupture them, if you apply pressure to them, you usually can stifle bleeding. In between those two major vessels, so we've got the going back to the heart low pressure and going away from the heart high pressure, right in the middle we have what's called the capillary network. So the capillary network are all these teeny tiny little vessels that are in the middle, these little vessels here, and they are very, very thin. They are only 1 cell thick to allow gas exchange to happen, just like the alveoli. They're under extremely low pressure. If you think about it, it's sort of like, imagine a really big river, and then the river splits up into 100 tiny little creeks, okay, tiny little flows of water. Well, the pressure in those tiny little flows are going to be much, much lower pressure than the pressure in the river where it started. And that's exactly what happens here. You get this distribution of flow into all these little vessels, and then the pressure goes way, 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 way down. So the pressure is much lower. It's 1 cell thick, and the blood cells, the red blood cells, actually travel through the capillaries in single file. So that's how small the capillaries are. And remember, red blood cells are among the smallest cells in your body. So we've got this structure that is like, it's only one cell thick. It's making a tube that's about 1 cell thick all the way around. And then we've got red blood cells traveling single file through that tube under very low pressure. If you watched the documentary already, you've seen this in action. That's what it looks like when they travel through single file. It's pretty cool. Okay, and those are the major differences here between arteries, arterials, arteries, capillaries, and veins. Any questions about any of this stuff? There are the questions that you're going to answer in the textbook. All this stuff, by the way, this in the next note is for tomorrow. To review what you got to do. Why does it keep starting this thing? There are the analysis questions right there. It's on 481 and 486. Questions? Everybody's very quiet today, not saying much. I get it. It's the first day back after a long weekend. Everybody's a little zonked, maybe. All right. I'm going to give you guys an opportunity to work on the questions, and if you need anything at all, please let me know. I'll be here the whole time.